So what we're talking about here in this next section is some of the tools that we use. So our class is really focused on biology, studying living things. And so we'll talk about some of the tools that we use in the science of biology, things like microscopes, and, spectrophotometers, things like that. I'll talk about it in a minute. So that's what we're going to talk about. Let's start with a microscope because it's one of the most important tools. Also one that you frequently will use in school, right? I'm sure you have before. But microscopes were really important because before there were microscopes, people didn't really know about very, very small things. They didn't know about tiny little organisms that live in the water. They didn't know that blood is made of tiny little cells. They didn't know that anything was made of cells. Because those things are so small, you can't see them without a microscope or some kind of tool. So microscopes were started to be sort of invented in the 1600s. And the first microscopes were pretty strange, and you might not recognize. Like the picture you have in the top right, that's a microscope. And this part here, this little, you can kind of see in this picture, there's like a little pin, and you put a sample of something on there, and then this hole is like a little tiny lens, and you would have to hold it up to light and look at it. And those are some of the very first microscopes. But they worked. People could see in great detail very small objects. So they started with like simple little lenses that people were grinding out of glass. That's the key thing that had to be sort of perfected or invented before microscopes could really be used. Some of the people, you don't need to memorize their names or anything, but I think it's kind of interesting to talk about the history. Anton van Leeuwenhoek, he made these really, really powerful lenses. He lived in like 1632 to 1723. He made these microscopes, but he was very secretive. He wouldn't tell anyone how he was making his lenses because they were the best around. But nobody knew how he could make these lenses. Only 9 of his microscopes are left that we have today. And these microscopes that he made way back then in the 1600s could magnify an image up to 275 times. So that was great for the technology that he had. He discovered bacteria. He discovered protists and vacuoles and muscle fibers, all sorts of different things. Okay, here's another type of like microscope from that sort of era. It looks more like probably what you're familiar with our microscopes, where you have a lens you look through, another lens at the bottom, and the sample goes here at the bottom. Another scientist was Robert Hooke. He wrote a book called Microscopes that actually, I thought I had a picture of it. I guess not. But it was this big, huge book where he had very detailed drawings of the things he was seeing under his microscope. He's the person that came up with the term cell for what he was observing under his microscope. So we use a couple different microscopes. The one you're going to be using today is called a compound microscope. I'll grab it for you so I can show you. This is a compound microscope. I'm sure you've seen them before. So in a compound microscope, you put what you want to look at on this place called the stage. Light goes through a little hole in the stage, through the specimen, up through a lens, gets bounced by a mirror this way, and then out through another lens that you look through to see the object. So in order to see something with this compound microscope, the object you're looking at has to be really thin because light has to actually be able to go right through it. And if I'm using this microscope, I can't look at my hand through this microscope because this light from the bottom is not going to go through my hand. All I'm going to see is black. Okay, so whatever you're looking at in the compound microscope has to be really thin. That's one of the limitations. Two different lenses, and this is, I think, something for you to write. Two different lenses magnify the image that you're looking at. One of those lenses is here. This is called the objective lens. It's the one you could rotate and switch back and forth. That's one lens. And then as the light, after it goes through that lens, it goes up here through the one in the eyepiece that's in here. That's another lens. So the object gets magnified twice. And that's one of the reasons it can give a high magnification. One of the things that you're sometimes asked to do is to find the total magnification of what you're looking at. To do that, you just need some simple math. You multiply the power of the eyepiece times the power of the objective lens. They're labeled. This one, for example, it says 10x. That means this lens up here magnifies the image 10x. The objectives are also labeled. This one is 4x. This one is 10x. This one is 40x. So if this is 10 and this is 40, if I have it set up this way, my image is going to be magnified 400 times, 10 times 4. Means what I see in here is going to be 400 times bigger than it actually is. Our microscopes can generally magnify something up to about 400 times. But there are other types of compound microscopes that can go even higher, 1,500 even. When you're looking at something in the microscope, because of the lenses and how they affect the light, what you see gets inverted, upside down, and also flipped backwards. So what you're seeing is sort of reversed. There's some blood cells shown under a compound microscope. Here's some onion cells under the compound microscope. Another type of microscope is the dissecting microscope. I have one of those over here too. So this is the dissecting microscope. It works a little differently. With this dissecting microscope, first you'll notice it has two eyepieces to look through. Sometimes it's called binocular eyepiece. Also, what you'll notice, once I plug it in, so it works a little differently. I just got to unwrap this cord. And what you have is this type of microscope uses a light that's shining from above. Remember, on the compound microscope, the light came from below. What that means is I could look at my hand through this microscope because light's coming from above, hitting my hand, bouncing up through the lens and out the eyepiece. So you could look at larger objects. Okay? It gives you, when you look through the two eyepieces, it gives you a three-dimensional view of what you're seeing. You could see depth very well. And you could see opaque objects, thick objects, objects which light doesn't go through. Typically, these microscopes don't magnify the image as much. They have lower magnification. For example, this one is 3x and 10x, so 30 total. So you get a different kind of view. Here is a B, for example. When you use the dissecting microscope, you get a three-dimensional view. Here's a slide. Now, when we do activities using the microscope, which we'll do a few of this year, what often happens is people have trouble finding and focusing on the object. So I'm going to tell you exactly how you should use the microscopes. First of all, you always want to carry the microscope with two hands. They are really expensive, so you don't want to drop them or break them. So if you have to move them from place to place, just carry it with two hands and be careful. If the eyepiece is dirty, you use special paper called lens paper to clean it. I have some of that if we need it. And when we are going to look at something, we always want to start on low power. That's the key thing. Start on low power. So when we're using the compound microscope, what we're looking at is usually on a slide, a little piece of glass, a rectangular piece of glass we call a slide. So if I want to use the microscopes we will be using have this special little thing called this mechanical stage, which is kind of cool. So you open up the clip, put your slide in, let the clip go, and it'll be held in place there. Once you do that, you can use these little knobs to move the slide left and right, or forward and backward. If you ever use the other microscopes, you kind of move the slide with your fingers, and sometimes you'll go way too far, and then you have to come back. These little knobs make it very easy to make small, tiny adjustments. So these are really nice to have these tools. Not all microscopes have them. So you start on low power. You have also a piece that's called the diaphragm. And the diaphragm controls how much light goes through the microscope. There's a dial over here, and it's got little holes in it. So if you select it for one of the big holes, a lot of light comes through. If you select one of the smaller holes, less light comes through. Because sometimes it's too bright. You need less light. Other times the image might be too dim and you need more light. So once you have your slide on, you're on low power, you start using the big knob, which is called the Coarse Adjustment knob. You look through, you turn this knob until you can see the specimen. You got to make sure what you're looking at, the little part on the slide, is right in the middle of the light. You turn the Coarse Adjustment until it comes into focus, and then you're good. Typically, you might want to go a step higher in magnification. After you have it in focus with this Coarse Adjustment knob, then you can focus with the Fine Adjustment knob. That's the small one. It moves at very, very small amounts. You want to see things even closer? Make sure what you're trying to zoom in on is right in the middle of your field of view of what you see through the microscope. And then you could switch to medium power. Once it's in focus and centered, then you could switch the objectives to medium power. It should be in focus still, or really close. If you need to adjust, again, you'll use the coarse adjustment knob. Focus it. If you want to look even closer, you could switch to the high power objective, the largest one, the blue one. Once you have it under high power, you only will use this fine adjustment, the smaller knob, because it's easy. If you use the coarse adjustment, you could smash into the slide with the objective lens. Sometimes when you switch to high power, you can't see it anymore. You lose the specimen. If that happens, you just go back to medium power, find it again, focus it, center it, and then try high power again. Just a couple other things. Sometimes we want to make a slide of something that's living, or a plant, or something else. We can make our own slides. Sometimes we have slides that are already made and glued together. We call those prepared slides. You don't have to do anything, just get the slide and look at it. Other times we want to make our own slides of something. In that case, we make what's called a wet mount slide. In a wet mount slide, basically, you take the slide, a piece of glass, you put a drop of water on it, you add whatever sample you're trying to look at, and then you cover it with a little piece of glass called a cover slip. It's like a little sandwich. Glass slide on the bottom, water in your specimen in the middle, and the cover slip over the top. And then you could bring the slide to the microscope and look at it. And that's how you make a wet mount. You'll be doing that today. Sometimes with cells, we want to add a special chemical that colors them a certain way. There are certain stains we can use. They make the nucleus stand out, or they stain the cell wall, and so forth. To do that, we just add them while we're making the wet mount slide. Now, like I already said, sometimes we want to calculate the magnification of our microscope. To do that, we multiply power of the eyepiece times the power of the objective lens. For example, 5x eyepiece, 40x objective lens gives us 200 times total magnification. But the more you magnify something, the less of it you see. The greater your magnification, as you zoom in, you see less and less of an area. We call that the field of view. Higher magnification, lower field of view. Lower magnification, higher field of view. The terms we have for the parts of the compound microscope, you studied in 7th grade. This is called the eyepiece up here. This is called the body tube. The part you twist to switch lenses is called the nose piece. These individual lenses are called the objective lenses. The flat part you put the slide on is called the stage. Some microscopes have clips, but this one has the mechanical stage like I just mentioned. The dial to change the amount of light is called the diaphragm, and then the light source is on bottom. You also have your focusing knobs. The larger one is the coarse adjustment that moves it a lot. The other one is the fine adjustment, which only moves it a small amount. And finally, when we look at things in a microscope, they're generally really small, obviously. So we sometimes use other units. A millimeter is usually the largest unit we will use when we're measuring objects in the microscope. Okay. When you look at a metric ruler, those small little lines, those are millimeter lines. Units smaller than millimeters that we'll use are called micrometers. There's 1,000 tiny micrometers in a single millimeter. We use this abbreviation here. This is the Greek letter mu. That symbol in M, that is the symbol for micrometers. So millimeters on this ruler is each tiny line. Okay. Micrometer is the symbol. And again, this red line is 1 millimeter. 1,000 micrometers are in that millimeter. So it's a really, really small unit. Sometimes you want to figure out how much of an area am I seeing in the microscope. And what you can do is you can look at a transparent ruler on the stage and measure it. If you put a transparent ruler, you can see how many millimeters across your microscope field of view is. You could also estimate how big objects are. If you know the field of view is a certain width, and you figure out how many things that you're looking at fit across, you can figure out how big each of them is. So for example, if this is my microscope, what I'm seeing, and the field of view is 2 millimeters, and I'm looking at some pollen grains, how big do you think this pollen grain is? Small, but if I know 2 millimeters is all the way across, how big is this one? Take a guess. It's kind of an estimate. Is it 1 millimeter? Is it this big? Or is it smaller? This pollen grain. What's that? Not quite a nanometer, because this, let's go in millimeters first. Right? If across this circle is 2 millimeters, this is 1 millimeter, this takes up maybe half of that. So it would be half of a millimeter, 0.5 millimeters. Okay? We know it's 2 millimeters across. Maybe 5 of these would fit across. That equals, about 400 micrometers, you could say. All right. Let's just finish up while we can. There's another type of microscope that we won't use. It's called the Electron Microscope. Instead of using light, like this microscope, it uses a beam of particles called electrons. And these are first used in the 1930s, and they can give a huge level of magnification, up to 10 million times. Here's like the first types, what they look like. When you're using an electron microscope, you have to coat what you're looking at in metal or other reflective material. So usually you can't look at living things in these microscopes. Here's a modern one. It doesn't matter about the types, don't worry about that, but they give different types of views. The one that creates some of the most interesting, I think, are the scanning electron microscope. Here's an ant with a picture taken through a scanning electron microscope. It gives a three-dimensional appearance. And it can get great resolution because the electrons are so small and have such a small wavelength. Our last slide I want to talk about is now beyond microscopes. But sometimes in science, we want to take a mixture of different molecules and separate them so that we can maybe analyze them. There's a couple ways we do that, and we're going to do both of these things later on in the year. One way to separate a mixture of molecules is through a process called chromatography. You have a solution with different materials in it. What we can do is we can put a thin piece of paper, suspend it in the solution, and let the solution soak up the paper. And you get a pattern that looks like this. As the solution rises up the paper, different substances separate out. This picture shows chromatography. If you took some plant leaves and smashed them up into a sort of plant leaf juice, and you did this process of chromatography, what you would find is that the different pigments that make up the leaves of a plant would separate from each other. There's more than just chlorophyll in leaves. There's all these other pigments, and you could actually see them and separate them using this process called chromatography. Another similar kind of process is called electrophoresis. We'll do that later in the year. This is when you put a sample of very small molecules, such as DNA or maybe amino acids. You put them in this gel-like substance. And then you add an electrical current to the gel. What happens is the molecules start moving because they're attracted to the charge. And they separate out by size. Small pieces of DNA or proteins can squeeze through the gel quickly and go far. Big chunks just go really slowly because they can't make their way through very easily. That's called gel electrophoresis. And it forms patterns like this. Each of these is a band of DNA that's been separated with electrophoresis. And then the last, we could also take an unknown sample of something and figure out what it's made of, what elements it's made of. Oops, we do that using chromatography. And chromatography basically uses how that element absorbs light to identify what it's made of. All right. So that's the end of our note.